So we'll also talk about other types of DNA-based damages. DNA-based damage through alkylation, oxidation, and radiation. So alkylation means, alkyl group can be added to your base structures. Okay, so for example, so some chemicals like nitrosamines can add alkyl groups to G base, like those spots could be typically alkylated, so extra alkyl group is added. So if that modification happens on G, guanine, then that modified G, it doesn't base pair with C anymore. Instead, it's going to base pair with T. So, in the DNA application process, of course, it's going to induce the mutation, right? And then reactive oxygen species like hydroxyl radical, superoxide, and hydrogen peroxide, and things like that can generate ionizing radiation, which can oxidize guanine to maybe 7,8 dihydro-8 oxoguanine. So oxoguanine, which has different structure. If that modification is made, then the oxo-G will pair up with A instead of C. Of course, that will induce the mutation if it's replicated. And then typically UV irradiation, strong light energy, can also damage DNA structure, especially if there are two thymines in a row on your same strand. Okay, and so those two thymine bases can be cross-linked through a strong covalent bond, so you can develop that odd structure, thymine dimer structure. Okay, if that thymine dimer structure is made, so that's a very odd structure, and then if that strand is used for your DNA replication template, then DNA polymerase doesn't know what to do with that, so it doesn't read, so it stops. It causes the stop of the DNA polymerase, so the replication would stop, so which is a big damage. So of course there are repair enzymes that can take care of this before the DNA strand will be used for DNA replication. We'll talk about that later on. And then gamma radiation, of course, radiation energy, and X-rays, okay, can cause double-strand breaks. Okay, so your double-strand DNA is broken in the middle of it, then it can cause extra damage. So that can cause the mutation as well. So there are many ways of getting your bases, their structures changed, and then those changed base structures will induce mutation if they're used for the template for the DNA replication. Okay. Also, there are some other mutagenic substances that can cause mutation. For example, base analogs. Those are kind of base-looking chemicals that have similar base structure, but they behave differently. Okay. So can cause mutation. For example, 5-bromouracil has a similar structure as uracil. 5-bromouracil has similar structure as uracil, but if base analog is accidentally added to your base, okay, then that 5-bromouracil will pair up with G instead of maybe T. Okay, so yeah, so the 5-bromouracil is analog to T because they have similar structure. T bears pairs of A, of course, but 5-bromouracil will pair up with G, so that's going to cause a mutation. Okay, and then intercalating agent, meaning these are the substances that can bind with the bases by going into the bases, two bases. Okay, so like they can stick well with the bases. Once they stick, they don't come up easily. So a good example is ethylene bromide that you use for staining your DNA. That's a strong intercalating agent. It goes and bind the bases and doesn't come up. If these intercalating agents bind on your bases, okay, they don't come up from your DNA base well. So if that happens, then if that DNA happens to be used for DNA replication as a template, the DNA polymerase doesn't know what to do, so they may delete or insert. The polymerase enzyme will do something strange around that area, so it's going to cause mutation definitely. Okay, so we looked at various ways of getting your DNA base kind of a structure changed, so that will cause mutation in your DNA. Then against those, I guess, conditions, your cells have a means of dealing with those DNA base changes. So there are various enzymes that can recognize and repair those gene damages. So this table shows you various types of repair mechanisms that are found from the living cell. So first, mismatch repair. So if mismatch repair is done, I guess a mismatch is made through DNA replication error by the DNA polymerase, okay, then these enzymes will come along, MOD-S, MOD-L, and MOD-TH, especially in E. coli. And then in human, like eukaryotic cells, there are similar enzymes with different names, and they can take care of the problem, okay, and they can repair that mismatches to the correct nucleotide. And then photo reactivation, light cause damage, and if you form like a pyrimidine dimers, then there are these enzymes that can repair those damages. And then basic excision repair, so let's say the bases are damaged, okay, then the damaged base part can be cut out. Then nucleotide can be replaced. And then nucleotide excision repair, again, if you have like a pyrimidine dimers again, or bulky adduction on base, then again these enzymes can come along and recognize those damages, and they can cut up and redo the polymerization, repair those. And double strand break, if you have double strand break on your DNA, again, these enzymes in E. coli can come along and repair those. Translesion DNA synthesis, pyrimidine dimer, a purinic side, a bulky on base pair, things like that. There are number of enzymes, DNA polymerases come along and fix those. Okay, so we'll take a look at those repair mechanisms. Number one, mismatch repair on replication error. So let's say DNA polymerase made a mistake. Then in E. coli system, okay, dimer of MOD-S, you see the MOD-S protein, two come along together, it's a dimer, okay, comes along. So they can recognize the mismatch because there's no base pairing made, so they are kind of hanging out. So the bulged or hanging out, distorted DNA structure can be recognized by those enzymes, and especially MOD-S can then recruit MOD-L, okay, which will activate MOD-H to nick the DNA strand near the mismatch. So that's what is shown. Okay, so MOD-L and MOD-H are shown up there. So they can be recruited, and then they can nick, meaning cut one of the two strands, especially the damaged strand can be cut. Okay, and then get rid of that part, and then redo that polymerization. Okay, so MOD-H selects selectively nick unmethylated daughter strand. So unmethylated meaning, the original template strand is methylated, but newly made replicated strain is not methylated yet. So that's what unmethylated is. So there's got to be a way to tell which one is the original, the template strand, which one is the, I guess, newly made strand with the error, right? So how do these enzymes recognize that? Because they can recognize that the original strand is methylated. So that's how they can tell which one they should fix. Okay. So unmethylated strand will be cut, and then UVLD helicase and exonuclease unwinds and remove the single strand with the nick beyond the side. Okay, so removing is done, and then the single strand gap is filled by DNA polymerase F3. Repair will be done by the enzyme, and then sealed by DNA ligase. Eukaryotic cells have similar mismatch system, like MOD-S and MOD-L, the homologous system, but they do not have MOD-H and hemimethylation. So methylation is unique to the prokaryotic cells. Okay, does it make sense? All right, and then since we talked about methylation, okay, so methylation is a modification of the DNA that happens after DNA, I guess, replication. So the enzyme called methylase, okay, can methylate A residues, okay, A residues on both strand, especially on the target site GATC, on that A, the base is methylated, okay. So but methylation takes some time. So if the replication going on and there's some time gap when only the template strand is methylated, but still the daughter strand is not methylated. Okay, so that's when this repair mechanism can take place. So during that hemimethylation time, so only one of the two strands methylated, the repair can take place. That's how they can recognize which one is the template strand and which one is the newly made daughter strand that with the mismatch, and they will repair the daughter strand. Okay, so that's what it shows. And then in terms of removing the daughter strand around that mismatch area, there are different exonucleases that can remove the daughter strand around that damaged, mismatch area from two different directions. Okay, so especially exonuclease 6 or RAGZ, they can remove DNA, the daughter strand DNA with mismatch from 5' ends, like this case. So nicking happens with mod H, and once that's done, these enzymes like EXO6 and RAGJ can come along and start removing the daughter strand from 5' end. So 5' end here, 3' end there, so 5' end here. So from the 5' side to the 3' side, so it's removing the daughter strand, including the mismatch area. Okay, and then polymerization will take place and we do the synthesis. And then here, and another enzyme, exonuclease 1, can remove from 3' end of the daughter strand. So see the nicking here, and then instead of cutting from 5' end, you can go from 3' end, because that's 5' end, so it's a 3' end of the daughter strand. You can go this direction. That's what it means. It's done by exonuclease 1. Okay, and then once that's done, polymerization will take place. That's what it shows there. And then there are these enzymes. They can take care of the damaged DNA right at that damaged spot. I want to talk about direct reversal of DNA damage by these enzymes. So first of all, the strong light, UV light radiation on your DNA, especially around the thymine, the covalent bonds are formed, especially 2 T's in a row, and that's a UV cross-linking covalent bond formation. Those are kind of strange bond formation, right? That's UV-induced damage, and that damage can be directly repaired by an enzyme that's called DNA photolyase enzyme. So the DNA photolyase enzyme is also activated by the strong light, and once the enzyme is available, it can come along and recognize the strange structure of the two thymines making the cross-linking, the covalent bonds, and you can remove that covalent bond between those T's and bring them back to normal structure. Okay, and another enzyme is it's called methyltransferase enzyme, which can fix the methylated guanine. So guanine could be methylated. Okay, that's odd structure, of course. Then that methylated guanine can be recognized by this enzyme called methyltransferase enzyme. So methyltransferase enzyme has SH, probably it's through its cysteine. Okay, it's going to take away that methyl group from guanine and put it on its own SH group. So it's going to transfer the methyl from guanine base to its own structure. So that's how it can fix it. So the SH from its own cysteine amino acid. So those are the two direct enzymes that can make direct repair. Okay. Also, there are another set of enzymes called glycosylase enzyme that can recognize the damaged base structures, and then when they recognize base structure, then they can cut up the base part of the nucleotide. So that's why they are called glycosylase, because they can cut up this glycosylic linkage. Okay, so the damaged base part is only cut up. So then you will have this odd structure. Phosphate and sugar is left alone only. So that's not a fully functional nucleotide either. So there are more additional enzymes come along and cut up the nucleotide. So endonuclease or exonuclease will cut that nucleotide up, and the polymerase will come along and repolymerize that place, and then the ligate that back. So that could be another way of dealing with the damaged base structure, cutting up the base and then replacing that nucleotide on that spot. Okay.